Just give me a second. Okay, we are live. So I would like to welcome one and all to this edition of the Wednesday Colloquium. And it's a pleasure to have Professor Gagandeep Kang joining us from CMC Vellore, currently actually from her home, I think. Just for everyone to be up on speed, I just would like to give a short introduction to the NSF Wednesday Colloquium series for those who are joining us for the first time. The NSF Colloquium series is as old as TIFR itself. It was actually first envisioned by Professor Homi Bhabha, our founding director, who actually would make a visit on every Wednesday to meet the faculty. And it also allowed for one of these interaction sessions at 4 o'clock, where all the NSF members, either a physicist, chemist, or biologist, get together with the director and listen to an eminent expert visiting TIFR on that day, and listening to a very important aspect of a scientific question that has been addressed by the expert. The idea behind this actually was to ensure that all of us get together under one roof for that one session. And that has brought dividends for the science and the quality of science in TIFR, both from the perspective of collaborations, as well as getting the young minds, the students, sort of trained to think about science, not just in a very specific part of a problem or a field, but to think about it broadly. So this training was very unique, and I think it is still maintained in TIFR. So we are extremely proud of this, and I'm very happy to host this session. Today we have, of course, an eminent expert in vaccination and vaccines, developing vaccines, Professor Gagandeep Kang. I would like my colleague from the Department of Biological Sciences, Professor Ullas Koltur, to introduce Professor Kang, and I invite Ullas. Ullas, could you please do the honors?
Yeah, thanks, Cherry. Of course, it's an honor and pleasure to introduce Professor Kang. Gagandeep is also known as Cherry to many of us. So I don't think she really needs an introduction. I guess anybody who is, mindful of the importance of public health and, science and data-driven policy that the governments often seem to not care about. I think Gagandeep has been one of the forefront of, dealing with many of these issues. But actually, Gagandeep started as a microbiologist, and she's a clinician, and she's been at CMC Vellore for most of her research career, I suppose, including as the head of the gastroenterology department. She also is the head of the Welcome Center, which is at CMC Vellore. She has, of course, been awarded by many honors and recognitions, including the FRS. But I guess what defines Gagandeep is not these recognitions alone. Her work in the context of infectious diseases, especially in the context of the GI, and her efforts at bringing to the fore the importance of vaccines and public health policies has been really something that has shaped our thinking of again using data-driven and evidence-based policies and research that needs a lot of boost in this country. So I guess I don't need to say more because she's very well known and her work is very widely popular. So Cherry, it's yours. Please do the honors.
Thank you very much for the introduction, Ullas. I'm going to be talking to you today about contagion and vaccines. And the reason that I wanted to discuss this is because it's topical, and because sometimes we get so lost in the details that we forget about the wider framing. How did we get to where we are? How are we doing? And what does this mean for the future? So if we think about contagion and about recorded history, even before there are historical records, we have been able to reconstruct some information on what has been happening with infectious diseases. One of the first examples comes from northeastern China, bordering Mongolia, a place called Hamin Manga, where archaeological discoveries published in a Chinese journal showed that in a collection of houses, where there is an aerial view of the houses shown on the left-hand side, there was one particular house that had almost 100 bodies inside it. What seemed to have happened was that men, women, and children, all up to the age of about 45 years, all their bodies had been collected into this one house, which was about 14 into 15 feet. So not a very large area. And this F40 house was filled with these bodies and set on fire. Now, what caused the infection? Why this was done? We have no clue. There is a reconstruction of the layout of the village, which is in a museum in Inner Mongolia. And what you can see on the right-hand side is when the discoveries were first made, all of the bones that were stacked up there. It also appears that in the early stages of the outbreak, there was an approach made to collect the bodies and lay them out in some order. And then at later stages, what happened was that everything was very jumbled together. So probably indicating that things got worse pretty rapidly. Now, while we don't know what caused that particular outbreak, from a similar tomb, which was at about the same time, about 5,000 years ago in Sweden, there is another collection of bones that were identified. And then from one girl, about 20 years old, from her teeth, the causative agent was identified. And this was Yersinia pestis, the cause of plague. Now, if we look at 5,000 years ago, most of our records are based on these kinds of investigations. But shortly after that, we started to have actual recorded history. And shown in the map on the right-hand side is the progression of the second pandemic of the Black Death in Europe. So over a period of years, you could see how the pandemic moved from 1347 in orange to green to yellow to purple to this pinkish salmon kind of color by 1351. You can see that this is a plague that arrives from the east and then slowly progresses northwards. Now, we've had records of the plague, particularly in the English language, where we had the plague spreading through the UK, leading to exoduses from the big cities. But we also have, much later, records of what was happening with plague in India. Especially once the British came in 1896, cases of plague began to be reported. And between 1896 and 1921, there were 12 million deaths. Unlike this hospital in Mumbai, the plague hospital there, most of the plague in India was actually a rural plague. And my great-grandfather was notable in being a rat catcher during the plague in the 1910s. He was actually rewarded by the British for it, and the way they used to reward people was to look at how many tails of the rats they had captured were submitted to them for compensation. There were other things that were done, and many of the practices of what happened during the plague in India were repeated for infectious diseases around the world. The stories are told of infected villages being evacuated or cordoned off. The idea that if you wanted to cross a cordon, you had to have a specific travel pass, and it needed to be certified by a higher level authority. In terms of the cleaning that needed to be done, and this was a very caste-based structure, where the lower castes were ordered to clean the floors and housing. And sometimes, buildings were made so that they could be repaired, but in other cases, buildings were abandoned if they had had multiple cases in there. Now, we learned to deal with these many forms of contagion, but the one that brought us to vaccines is also a fascinating story. Now, smallpox is a disease that the older individuals, perhaps parents of everybody who is here, would have encountered and known people with. I know that when I was in school, it was still pretty common to see people who had the pockmarks of smallpox, a very characteristic picture that was common in many people in India. Now, this was a disease that has been described for a really long time, but if we look at the history of what has been recorded, even in the 1700s, where populations were much smaller than they are today, in Europe alone, about 400,000 people died every year of smallpox. It was a desperate situation, as bad as the stories that are told of the Black Death, of people trying to throw out anybody who was potentially infected. The same thing would happen for smallpox. So when we think about both Benjamin Jesty, who came before Edward Jenner, but is not as well remembered in history, and the work that Edward Jenner did, those we don't understand the scale of relief that these interventions brought to the population. If we look at Jenner's experiment, Jenner didn't know what caused smallpox. He did know of variolation, which was a practice that had been introduced from China to India to Turkey and then into the UK, which was to take the scabs of people that had had smallpox and were healing, to powder those scabs and then rub them into scratches in the skin in the hope of inducing a milder form of disease, which would then subsequently protect that individual from smallpox. So that practice of variolation had been introduced almost 100 years previously into the UK, was widely used, but was also a very chancy affair, because if it worked well, it was great. But in some situations, what would happen was that the person would develop smallpox and die because the variolation itself led to severe disease. So Jenner's experiment is important because it is a way of making vaccines that is to some extent followed even today. And this is called the Generian approach. What he did was to take a material that was from a related virus. Didn't know it was a virus, just knew that it was a related kind of disease. He took material from a related virus and gave that to his gardener's son, James Phipps, 8 years old, was inoculated with the cowpox. And then once he had recovered from cowpox, he was challenged with the smallpox virus. So deliberately tried to induce a smallpox infection. And then when James did not develop smallpox, that was proof that the cowpox was protecting against smallpox. Now, this was a discovery that gained prominence very, very quickly because it was finally a safe solution to a problem that was facing all of humankind all over the world. Within 4 years of Jenner describing what he was doing with the cowpox virus, vaccines were being given across Europe and North America, and smallpox was eliminated from much of the industrialized world in 100 years. Now, if you are interested in history, there's plenty of history between 1800 and 1900 of how smallpox vaccines began to be used and made in different parts of the world. In the original approach to vaccinating Northern America, they actually took cows that had cowpox, put them on a ship and sailed them across the Atlantic so that they would have a supply of cowpox that they could then use for the vaccine. Now, it took us until 1979 to eradicate smallpox. And if you think about it, it was really only 20 years of a global eradication program when the world decided we were going to eradicate a disease and to the point where it got eradicated. Why have we not been able to replicate that success? Why were we able to do this with a vaccine that was made in 1796 with all of the tools and all of the technologies that we have today? Why have we not managed to eradicate another infection? I think it's very important to understand that there were certain special characteristics of smallpox that led to the eradication of smallpox. The first and the most important one was that if you had smallpox, you couldn't hide. The smallpox lesions were so evident that it was possible to identify all cases of smallpox. You didn't need a test to identify a case. You could take a look at the person and you could identify that they had smallpox, which meant that it was possible to train people to visually identify all cases of smallpox and bring them to the attention of the authorities. The second important feature was that there was a lack of reservoirs. Smallpox is caused by a virus. It cannot hide in the environment. It can only be inside a living organism. And for smallpox, there are no animal reservoirs. Smallpox is or was a purely human infection. So transmission of smallpox had to be human to human. It could not be a zoonotic infection. The virus could not go and hide in another animal species. And then you had to have effective tools. And one of the things that was very effective was the vaccine. So for smallpox, if you had gotten smallpox and you recovered from it, or if you got the vaccine for smallpox, you had permanent immunity. You never got smallpox again, infection or disease. So a person who was vaccinated or a person who was infected with smallpox would never again get infected and would never again, therefore, transmit disease. So this led to a situation where now it became possible to think about vaccination strategies that were subsequently thought about for SARS-CoV-2 as well, which were called ring vaccination, which meant essentially it was not necessary for the entire world to be vaccinated. All you needed to do is to identify all cases and around those cases create a ring of vaccinated individuals so that that case could not then transmit disease to others. So this ring vaccination strategy essentially meant that as long as you could detect all cases early and you could vaccinate all contacts of that case, you would be stopping the onward transmission of the virus. And over time, the virus would essentially die out in the population because it had no hosts to get to. That's exactly what happened with smallpox. And after that, I think we forgot some of the lessons of smallpox and we decided we would eradicate other viruses. We've been unsuccessful because we forgot those lessons, and I'd be happy to discuss that at a later stage. Now, the smallpox vaccine was developed without us knowing anything about the organism or knowing how it caused disease. We did finally get to the epidemiology and the science in formulating the strategy of ring vaccination, but it did not require, at least initially, the kind of information that was needed for subsequent vaccines to be made. And for that, we have a few people to thank. The first, of course, is Robert Koch, who is remembered, of course, because he identified Mycobacterium tuberculosis as a cause of TB. But he also formulated Cox postulates that then led to an explosion of understanding in infectious diseases. And Cox postulates essentially were that if you have a disease, there must be a microorganism present in every case of that disease. You should be able to isolate that organism. And if you put that organism into a healthy host animal, then you should be able to replicate the disease in that host and be able to re-isolate the microorganism once again. So this was Cox postulates. It was used to identify a range of pathogens and their association with disease. And Koch actually used that information to think about making vaccines along with others. He traveled the world, and a map of his travels and some of the costumes he wore are shown here. But he went looking for microorganisms in order to be able to make vaccines. And one of the first human cholera vaccines that was made by Robert Koch on his travels to the Middle East. Now, cholera was, of course, a disease that caused pandemics and spread around the world quite easily, traveling in people, again, as a human limited disease. But the science of epidemiology and the science that Robert Koch and other microbiologists had worked on really came together in the third pandemic of cholera, where in London, John Snow was able to show that there was a clustering of cases that centered around Broad Street in central London. And he, through doing a series of maps that related where the cases were with their water supply, demonstrated that it was a pump in Broad Street that was leading to a contaminated water supply that was killing people because of cholera. Very famously, John Snow removed the handle of the Broad Street pump, and that brought the cholera outbreak in London under control. Now, this was the first bringing together of mapping strategies with identification of potential sources that has informed infectious disease epidemiology since then. In addition to the work that was being done in epidemiology, we can't talk about vaccines without talking about the person that gave vaccines their name. Louis Pasteur called them vaccines in honor of the work that Edward Jenner had done. And he was responsible for making the first live attenuated vaccine, and he made it for chicken cholera. He had identified, using Cox postulates, that chicken cholera was caused by Pasteurella multocida. He cultured it in 1877, and through serendipity, because Pasteur went on holiday, his assistant went on holiday, they ignored a Pasteurella multocida culture that was left on a bench. And when they came back and tried to infect chickens with the Pasteurella, they did not develop disease. And then Pasteur thought about it and challenged the chickens with a fresh culture of Pasteurella multocida and showed that the chickens were protected. He therefore surmised that exposure to oxygen had weakened the bacteria, converting them into what is now considered the first live attenuated vaccine. Now, if we think about where we are today, one of the things that we have to recognize is that because we are changing the world, we are changing how diseases happen. And it's important for us to recognize that the future only holds more infectious disease. SARS-CoV-2 is one in a really long line of infectious agents. And as we travel more, as climate change happens, deforestation happens, there are just more people in the world, we are going to continue to see more infectious diseases emerge. If we are lucky, many of them will restrict themselves to outbreaks and epidemics, which are more localized rather than pandemics. But if we look at pandemics and what causes pandemics, most of the large outbreaks that travel very easily are caused by viruses. Since 2000, we've had the 3 coronaviruses, but we've also had H1N1, Ebola, Zika. And if we look at the global risk report published by the World Economic Forum, then infectious diseases rank number 4 by likelihood, but number 1 by impact. And that's really been borne out over the last couple of years. Now, I'd like to take you back to discuss one particular outbreak that resulted in reshaping our thinking around outbreaks and vaccines. Now, Ebola in West Africa, Ebola has been known since 1976. Mostly Ebola used to happen in remote forest villages in Africa, and the same principles that I described to you of isolating villages, of making sure that contact was prevented, were being followed, and most Ebola outbreaks tended to be small. In 2014, 2015, what happened was that Ebola came into cities in West Africa. And when that happened, the numbers of cases really went up. We wound up with almost 30,000 cases in that outbreak finally. And it was mainly localized in 3 countries, Liberia, Sierra Leone, and Guinea. Now, the interesting thing about Ebola was that we had no treatments and no vaccines at the time that the outbreak occurred. But when global authorities started to look around, they found that there was a vaccine candidate that was sitting in a freezer. There were also antibody candidates, monoclonal antibody candidates that could be considered for therapies that had never been evaluated before. So a decision was made to try and evaluate these therapies and prevention strategies. But unfortunately, by the time WHO and public health agencies and countries and everybody got their act together, the measures that had been taken for the control of Ebola were already taking effect, and the number of cases had declined a lot. So if we can handle outbreaks like SARS-CoV, H1N1, Zika, Ebola without vaccines, do we actually need vaccines when other approaches work? How did we handle SARS-CoV? How did we handle H1N1? I think some examples like this are useful to understand. SARS-CoV, rather like smallpox, only the person with that infection only became infectious when they already had symptoms. So SARS-CoV, you would develop respiratory symptoms and fever and then become infectious. So that makes it easy to identify a person with infection and isolate them. And SARS-CoV was handled by isolation and strict infection control practices. In fact, to the extent where we reduced the reproductive number and the disease disappeared. H1N1 wasn't like that. H1N1 was a disease that could infect people. They could be asymptomatically infected and could transmit disease, and you would not know without testing that they were infected. So how did we manage H1N1 with only the developed world getting vaccinated and low and middle income countries barely receiving any vaccine? Well, it turned out we got lucky. H1N1 was a very mild infection. And this is a virus that has now become an endemic virus that is part of the flu viruses that we see every year. What about with Zika? Zika came in, burnt through the populations, could cause asymptomatic infections, also resulted in serious consequences for women who were infected for the first time during their pregnancies in microcephaly and other conditions in their newborns. But Zika essentially burnt through the population so quickly that by the time it came to evaluating a Zika vaccine, everybody had already been infected. Ebola was handled during the first outbreak by very, very strict infection control and by isolation practices. So we managed. And the important thing to understand is that we need to understand transmission, we need to understand severity, but every infection is unique, and without understanding epidemiology and pathogenesis, we have very little hope of control. For Ebola, however, vaccines that were evaluated in the 2014-2015 outbreak and subsequently have turned out to be a great success story. Last year, the Democratic Republic of Congo had its 13th Ebola outbreak, and unlike previous outbreaks, it could be cut off quickly with vaccination. They had only a total of 11 cases, but they were able to rapidly do a ring vaccination strategy, and that stopped Ebola in its tracks. So vaccines can make a difference when it comes to handling outbreaks. Now, if we look at the technologies that have given us vaccines, I talked to you about the Generian approach to vaccines, where you take an animal virus and you use it for a human disease. But the other vaccines that have been developed from Pasteur to Hilleman followed a technology that went from 1796 up until the 1950s, that resulted in either inactivated or live attenuated vaccines, and all of them were based on the principle of isolate the organism, inactivate it or attenuate it, and then inject that into people. So this resulted in a number of bacterial and viral vaccines, many of which, all of which, we continue to use today. But after the 1950s, we've had a lot of new technologies that have advanced vaccines beyond the older approaches that we had. In 1950, we got cell culture, and that allowed us to make a number of viral vaccines that it had been impossible before that. In 1980, we had recombinant DNA technology that now allowed us to make protein vaccines instead of relying on the whole organism. We could now make safer vaccines. In 1990, we got glycoconjugation, which meant that vaccines that had earlier been limited because they were based on polysaccharides could now be conjugated and result in a robust immune response and be used then to address diseases in children who could not be immunized with the polysaccharide vaccines. And in 2010, we got reverse vaccinology, where working from the sequence of an infectious agent, you could identify what was likely to be the most important immunogen, and then use that approach to be able to develop vaccine candidates, evaluate them in animal models, and then take them rapidly into humans. But really, in the last 20 years, new technologies have really exploded, and as has vaccine development. We've had reverse vaccinology, we've had, following that, structural vaccinology. We now have synthetic biology that has allowed us to make the RNA and DNA vaccines. We have all kinds of new adjuvants, and we have the ability now to deeply interrogate the human immune response in order to identify which antigens should be selected for vaccine development. Now, with SARS-CoV-2, we've been very fortunate in that every platform it is possible to have for any kind of vaccines has been utilized to make vaccines for SARS-CoV-2. So on top, we have the live attenuated and inactivated vaccines that use the entire organism. And then we have the products of recombinant technology, which can give us virus-like particles or just proteins. And then we have vectored vaccines with a range of different kinds of vectors, and DNA and RNA vaccines. Now, the big thing that has changed in the last couple of years is that we've actually managed to make vaccines in 10 months. Prior to this, the fastest vaccine that had been made was the Mumps vaccine, which was made by Maurice Hilleman in a period of 4 years. He wanted to make a mumps vaccine, came home one day to find that his daughter had mumps, and swabbed her, took the swab back to the lab, grew the mumps virus, and made a vaccine out of it. Took him 4 years, but we still use the mumps vaccine that came from Gerald Lynn Hilleman. Now, usually when we think about vaccines, we talk about $500 million and about 10 years. What has happened in the last 2 years is that a lot of things have changed in terms of how we approach vaccine development and what we have been doing in order to make sure that we can develop vaccines as quickly as possible. So early stage development, technology transfer, looking at how we can shorten manufacturing times and what we can do with regulatory processes to make sure that vaccines can be made quickly in an emergency. But I think the important thing that we need to remember is that we were able to move much faster than we had anticipated because of two major reasons. One is all the advances in vaccinology in the past 20 years. And the second is organizational preparedness following Ebola. So while many of us tend to think about the science, and the science is critical and important, really to get vaccines into people requires manufacturing and administration, which are equally, if not more important than the science of vaccines. Where are we today? We are in a situation where vaccines are being made and given out at an unprecedented scale. I believe about 35 million doses of vaccines being given every day. And in India, we are in a fortunate situation of having vaccines that are made in this country, which has allowed India to be one of the countries that is really doing much better than the global average in vaccinating its populations. Obviously, we have a ways to go, but in general, we have done very well for both production and for delivery of vaccines. Now, we hear a lot of hype about vaccines and how the vaccines are doing or not doing in the media, but I think it's important to remind ourselves that when WHO established a target product profile, what it was looking for from a vaccine that had public health impact was a vaccine with approximately 50% efficacy that gave protection for 6 months. And I think that where we have clean and clear data, that is absolutely the case, that most of the vaccines that we have are providing a good level of efficacy against severe disease and mortality, and for longer than 6 months. Now, I'd just like to take you through very quickly the vaccines that we have today against SARS-CoV-2. The inactivated vaccines, the first ones off the block were Sinovac and Sinopharm. And these vaccines are probably among the globally most widely distributed vaccines in the world, because the companies that manufacture them began to supply them very early to South America, to Africa, as well as to their own population in China. India's Bharat Biotech's Covaxin has been used in about 11% of our population so far. 11% of the doses given in our population so far have been really of Covaxin. It's now approved and exported to many countries and has been approved for children, and that vaccination program has also started. If we look at the protein subunit vaccines, these can be virus-like particles, or they can be protein subunits. If they are subunit vaccines, they need to use an adjuvant along with them.
Professor
And
Kang,
yes,
sorry, interrupting you. Could you go back one slide back?
sure.
So just quickly, just a summary. So when you say inactivated vaccines, which means it's just dead virus that sort of
Pretty much. It's so you can kill a virus by treating it with heat or with chemicals. You may, well, since viruses aren't alive, you don't kill them,
yes,
but you can treat them either physically or chemically and make them incapable of replication. So
correct.
one of the reasons that it's really complicated to make inactivated vaccines is because you need to show that every last virus in the preparation that you have that is being given to people has been inactivated. So the testing for inactivation, ensuring that there is no risk of the person developing the disease because a few live viruses sneaked in there, is the challenge in making inactivated vaccines. So it's an old technology, but there are issues with the manufacturing.
Okay, and the idea is essentially to generate some antibodies against it. The body has to generate just by looking at the outer coat of it, but it doesn't know whether it's capable to be produced or not, but it just generates those.
That's correct. So
and that's okay.
But in the case of the inactivated vaccine, because you are presenting more proteins than just the spike protein, you anticipate that you will have antibodies to other components of the virus as well.
Oh.
Now, does that matter? Potentially, it could matter. We know that where the antibody response is concerned, the spike response is sufficient. But for T cell responses, it has been argued that having other proteins could make your T cell response more robust, more broad across variants.
Right.
So that's why there's also a focus on continuing to develop live attenuated viral vaccines.
Okay, thank you. Thank you very much. Thank you.
Okay, so virus-like particles are basically proteins. They can fold to look like viruses with a similar sort of structure to viruses in some cases. In other cases, they look like viruses, but the structure is different from the original virus. So these vaccines, examples of those that are available in India include Novavax or Covovax and the Corby Wax vaccine made by BioE. The Covovax vaccine is very interesting because it uses an adjuvant called Matrix M, and that is an adjuvant that has not been used in any commercially licensed vaccine previously. Corby Wax is just the protein subunit in the receptor binding domain, and that uses an adjuvant that is called CPG. And the Corby Wax vaccine is similar to a vaccine that's being made by a Chinese company called Clover. And that vaccine has just had its phase 3 efficacy results showing very, very high efficacy and good protection against a range of variants up to Delta. But the results came out before Omicron. mRNA vaccines, of course, everybody knows about the Moderna and the Pfizer mRNA vaccines, but I'd just like to highlight that in Pune, Genova is making an mRNA vaccine that it hopes that it will be able to deliver at room temperature. As you will be aware that cold chain requirements for the mRNA vaccines have been something of a challenge in some countries where these vaccines are being used. So Genova's results are expected any day now, and then India might have an mRNA vaccine. mRNA vaccines, DNA vaccines, and viral vectored vaccines all work essentially the same way, where they deliver the message for the spike protein to the host cell. The host cell then becomes responsible for making the spike protein, and then the body recognizes the spike as a foreign antigen and responds to it. Of course, mRNA vaccines, these are the first vaccines that have been made using this technology, and there were two kind of approaches that made these vaccines feasible. The first was the modifications of RNA that allowed for the mRNA to increase the yield of the protein that is made inside cells. And the second modification, which resulted in a suppression of the inflammatory response that's usually made when an RNA molecule is introduced. So that was the RNA technology itself. The second important component was the synthesis of the lipid nanoparticles that protect the mRNA until it can get inside the cell. Without advances in either of these two technologies, we wouldn't have had mRNA vaccines today. Viral vectored vaccines, we have three that are widely available. The ChAdOx vaccine, made also by the Serum Institute of India, Covishield, the Gamaleya Sputnik, and the Janssen and Janssen vaccine, which is also being made in India by BioE. All of these are adenovirus vectors derived from animals or humans, and all of them carry the message for the spike protein. In all of these cases, the spike protein is the full-length protein. So again, similar approach, deliver message, cell makes a spike protein, body recognizes as foreign, makes antibodies. The advantage of the viral vectored vaccines is that the viral vectors themselves are supposed to induce a better T-cell response. And in studies that have been done on volunteers, it does appear that both mRNA and the viral vectored vaccines induce a strong T-cell response that is maintained over time and is much more broadly reactive than the antibodies are. We don't yet have live attenuated vaccines, but there is work that is ongoing with Serum Institute and Indian Immunologicals, where the idea is to take the genome of the virus and to detune the genome so that it, basically it reduces translation efficiency. So less protein is made, and that results in a virus that is unable to cause disease, but induces an immune response. DNA vaccines, of course, the first licensed DNA vaccine in the world is the Zydus Cadila vaccine that is delivered through a device that's called PharmaJet. There are also US and Japanese companies that are working on DNA vaccines. So these are supposed to enter the program fairly soon, but it has not happened yet. So that's a quick summary of how we got to where we are. And now just wanted to highlight the kinds of things that we need to think about for the future. Obviously, top of mind for everybody is variants. And to that, I'll say we need to be thinking about variants with more data than we have at this time. There is work already being done. There are vaccines already being developed, but it's important for us to understand the breadth of protection and the duration of protection for the future. There is work that is ongoing to look at how we optimize the immune response to make sure that we have the best possible, longest lasting protection possible. And then we have to think about what it means if some of the world gets immunized and the rest does not. Some of this is due down to vaccine inequity at the moment, where there just hasn't been supply for the vast majority of the population. But through 2022 and 2023, that's going to decrease. And a bigger problem is going to be the people who don't want to get vaccinated. And then, of course, if we've learned anything in the last 2 years, we've learned that we know very little and we should expect the unexpected. I just wanted to make one point about variants, that variants keep arising all the time. And for vaccines, they mostly haven't mattered so far. Will they really matter for the variant we have now or the ones to come in the future? We'll have to wait and see. If you will remember, when the Delta variant, which is shown here in the many shades of green, came around, we were worried that vaccines would fail, and early data from the UK seemed to indicate that protection against Delta was less than against Alpha. But we managed pretty well with the vaccines. Now in purple, we've got Omicron. India has Omicron shades, BA1 and BA2, and we still need data to see how well or badly the vaccines are performing in our setting. Now, as a global community, it's been a phenomenal couple of years. You know, the Mrna vaccine was designed in 2 days and produced in 6 weeks. Today, we are in a situation where Pfizer is expecting to have in March 2022 a combination vaccine that includes Omicron. So people are thinking now that we can have a vaccine in 100 days when we have the next outbreak. But will a vaccine be required for every outbreak? Will a vaccine solve every outbreak? We really don't know yet. It's possible for many kinds of viruses, but not for everything. And we certainly don't know what the unknowns are. So with that, thank you very much for your attention, and I'd be happy to take questions.
Yeah, thank you very much, Professor Kang. That was very nice. And considering that you are also infected right now and you're still delivering the talk, I really am, this is a heartfelt thank you. After this wonderful talk, I request, so there are a few questions already in the chat box. I request Professor Sandhya Kaushika, who has asked 2 questions, to please unmute herself and then maybe ask those questions directly to Professor Kang. Sandhya?
Hello, Professor Kang. As usual, wonderful talk. So my first question is, what goes into making a vaccine that gives sterilizing immunity? And is that a possibility for either the corona class of vaccines, viruses, or the flu class of viruses? And why is that not talked about more?
So the concept of sterilizing immunity is one where you are so protected that you never get infected again. And I have not really seen that happen for anything that is a mucosal infection. So I know we live in hope of sterilizing immunity for influenza, for SARS-CoV-2, but really there is no mucosal infection for which we have managed to achieve sterilizing immunity. Take, for example, polio, which is a virus where we have been enormously successful. But with polio, if you give vaccines, what you essentially do is when a person makes an immune response to a polio vaccine, they will never develop polio, the disease, but they can develop polio, the infection. They can carry the virus for a while in their gut. And that's the best mucosal vaccine we have at this time.
What's rate limiting for that? Why is it that we are not able to get there?
Well, we don't understand. So with every pathogen that results in infection of the mucosal tract, asymptomatic infections are quite common. So the first time you see it, you'll get infected, you'll get sick, and then the next time you won't get sick or you'll get less sick, and over time you will build up enough local and systemic immunity that you don't get sick at all. I wonder whether we just think, or our immune system just thinks that it's not worth the effort to shut it out completely, right? You have to have some balance in life, and if you're not getting diseased, why go that extra mile?
I see. So my next question was about epidemics, like the Zika epidemic was sort of self-limiting without a vaccine strategy. And how common is this, and what leads to this sort of burnout of certain epidemics? And this is, of course, going to in turn relate to how pandemics also die out. Your thoughts on that, greatly appreciated.
So I think with, we talked about mucosal infections. And mucosal infections are your gut, your respiratory tract, your genital tract, right? So those are areas where permanent protection from infection is very hard. And if you talk about the rest of the body, now you're talking about sterile sites. You do not want bugs in your bloodstream. You do not want a bug in your liver or your brain, right? So here is a situation where you want to create sterilizing immunity, where you need to have an immune response that shuts down the infection as soon as it happens. So the most successful immune responses are the ones that encounter a virus once, and then the next time it comes around, it is not allowed to replicate within that sterile space. An example of that is measles. So for measles, if we vaccinate, we create a sterilizing immunity. An example of where an infection creates sterilizing immunity might potentially be chikungunya, where if you get chikungunya once, you won't get chikungunya again because you made a good immune response to it.
Okay, thank you.
Thank you. Thank you, Professor Kang. I'll request Diksha to unmute herself and ask the question.
Yeah, thanks, JD. Nice talk, Professor Kang. So I was wondering, I have a couple of questions actually. For mRNA-based vaccines, instead of having the mRNA just for the spike protein in case of SARS-CoV-2, is it not possible to have a soup of mRNAs and then subject the vaccine to the subject so that we can again have immune response for multiple components of the pathogen?
Absolutely. So multi-component vaccines are a thing. And for dengue, that's what we are trying to do, to make vaccines that are for all the 4 types of dengue. For SARS-CoV-2, it is possible to make a vaccine that has different kinds of spike proteins. Let's say you want to make a vaccine. So the Pfizer vaccine is an example, where they're planning to have the mRNA for what they have existing as a vaccine and add the Omicron spike sequence as well.
Okay. So the next thing is to deal with variants like Delta and Omicron, etc. The vaccines which are based on attenuated viruses, since again they trigger an immune response for multiple components of the pathogen, they should be able to take care of the variants. Then why are these vaccines not preferred, and why are we failing them, though we have such vaccines?
Well, the thing is, they may give you a broader based response, but which are the most efficacious vaccines? Okay, so how much disease is actually reduced by using one class of vaccines versus another class of vaccines? And in the early data on inactivated vaccines from Sinovac and Sinopharm, the protection has not been maintained very long and has been lower against the virus itself and against variants than other classes of vaccines. Will live attenuated vaccines do better? We won't know until we evaluate them.
Okay, thank you.
Thank you, Diksha. I request Amol Dighe to ask his question. He typed it in chat a while back. So Amol, please.
Yeah, hi. So see, now of course, we can in some sense test and know that the virus has mutated. I suspect that, I'm not a biologist, okay, so I suspect that if you go 1 century ago, it would not be possible to look at the virus and see it has mutated. So have there been records which show that people had realized that it has mutated, maybe because of change in symptoms or something of that kind? And other records that show that the vaccines that were made for earlier virus did not remain as effective because of this mutation?
Well, if we go 100 years back, or 100 and a little bit years back, when there was the influenza pandemic, right? Or at the end of the First World War, they actually thought that it was caused by Haemophilus influenza, which is a bacterium. It took until 1931 when they were able to grow the influenza virus in embryonated chickens' eggs that they realized that this was not a virus that was causing the infection. Now, in terms of studying the relatedness of strains, there are all kinds of ingenious mechanisms that have been used by enterprising virologists and disease biologists. We actually have the sequence of the 1918 H1N1 influenza virus, because it was recovered from a grave in Alaska where everybody in a settlement had died of the influenza virus, and people remembered where the graves were. So it was possible to go back and sample the lungs of those individuals because they'd been buried in permafrost. They were able to recover and sequence that virus. So studying strain variation is being more of modern technology. But as I mentioned, what was Louis Pasteur doing? Louis Pasteur was looking at patterns, and essentially what he found when he decided to use different strains for the development of vaccines or evaluation of what was going to become vaccines for anthrax, for rabies, for chicken cholera, which I described earlier, was to actually characterize those in terms of their biological behavior.
Okay.
Thank you.
Thank you, Professor Kang. Thank you, Amol. Now I request actually Vijay. Vijay had raised his hand and unfortunately his hand went down. I don't know. So he's back here. So Vijay.
I thank you for a wonderful presentation, Professor Kang. My question is not related to vaccines, but to more about the spread of viruses. And, throughout this pandemic, right from the beginning, it started with fomite transmission being the dominant source. And then it took a long time for the airborne nature to be sort of, made to be the prominent method of spreading. So what I wanted to ask you is that, does it really depend on the type of the virus? Or is it safe to assume that a respiratory virus is going to have airborne transmission as the most dominant mechanism? And why was this so confusing at the beginning? Is this really something we are only learning now? Or is it just a situation of bad communication?
So according to the aerobiologists, it's because the people who deal with infectious diseases don't talk to them enough. And this classification, so when I was a medical student, we were taught this classification of large droplet versus small droplets and aerosol transmission as a matter of, this is how viruses travel around in people. We now know that that is not true. And some of this was down to misunderstandings that are very hard to trace exactly how it became accepted fact. You can get up to a certain point and see where the story is leading, and then there's a gap and you don't know how this fiction was maintained and became accepted completely. But there is much better experimental evidence now, and that seems to indicate that with any virus, you're going to have to worry about aerosol transmission. One of the reasons why specifically for SARS-CoV-2 it was accepted so easily that it was large droplet transmission and therefore you needed to pay attention to surfaces was because SARS-CoV-2 among viruses is a very large RNA virus. But we have to remember that a large RNA virus is actually very, very, very small.
Thank you.
Thank you. Thank you, Professor Kang, and thank you, Vijay, for asking that question. Maitreyi is next. Maitreyi?
Yeah. Thanks for a wonderful talk. I was wondering what so there's been work that suggested that the Omicron variant causes less severe disease, and there's some literature showing that. I wondered what your thoughts were on why you think it's more contagious. What makes it more contagious?
So right now, the argument for greater contagiousness is that it's a very upper respiratory virus. So that means that it's located where it's easy for it to get into respiratory secretions and get transmitted. And it also has a higher viral load. So the more the virus you excrete, the greater the number of particles there are floating around that allow for people to get infected. If you remember, the early arguments used to be 15 minutes of face-to-face contact with an individual. And now it's down to, well, you can still get infected if you enter a room 3 hours after an infected person has left it. So that doesn't leave us with very many places to hide.
Okay.
Yeah. But you also think that the fact that it's not going deeper into the respiratory tract allows viruses to accumulate higher up, and so simple things like just breathing,
talking will lead to a lot of expulsion of virus.
Right. Thank you.
Thank you. Thank you, Professor Kang. Thank you, Maitreyi, for asking that question. Ullas is next.
Yeah. Hi, Cherry. So just continuing on that topic itself, is that is it likely that is there any evidence to indicate that there are other receptors besides ACE2, and given the mutations that we have, so is ACE2 binding is also, because we know there's an immune arm, so how much of that is also dictating the Omicron disease outcome? So is there any evidence to indicate that?
So SARS-CoV and SARS-CoV-2 both use ACE2. The other coronaviruses don't necessarily have the same kinds of binding to ACE2 receptors. But Omicron is different in that it doesn't seem to be dependent on the second molecule, Tempress. It has a different pathway of entry, uses endosomes. So Omicron behaves so differently in terms of how it infects, where it localizes, its higher transmissibility. Sort of, there isn't enough mutations in Omicron to call this SARS-CoV-3, but it's certainly headed in that direction.
Okay. The last question on that, since we brought up the SARS-CoV-3, is that particularly when, in terms of severity of the diseases, do you think for a zoonotic infection, if a population is priorly exposed, do we have enough information on either enhanced or decreased susceptibility because of a skewed host response? Is it likely even to think about this, or?
With all zoonotic infections, the biggest thing is how well the virus is going to replicate in the host. Zoonotic infections happen all the time. Reverse zoonotic infections happen all the time. But because most infectious agents, particularly viruses, have evolved to be specially adapted to a host, then you don't see a lot of cross-species transmission. So it takes a pretty special virus to be able to cross that species barrier. And in SARS-CoV-2, we have a virus that is very promiscuous where species are concerned. May have come from bats, but it seems to go into all kinds of animals and come back again quite easily. We've seen that with mink. We've seen that particularly in the cat family. A limited amount of transmission that goes into dogs. Don't see the reverse of it coming back from dogs. And it's one of the hypotheses for why we have Omicron, that the mutations may have arisen from a virus that crossed into animals and back again. Oh. Now, are these all general principles for this virus or all viruses? I don't think that we can say that in terms of either the mutations that happen or our immune system's ability to handle these viruses.
Okay.
Thank you. Thank you, Ullas. Thank you again, Professor Kang. Professor Gautam Mandal.
Hi, it was a wonderful talk. I wanted to, so I'm not a biologist, first of all. So I read in many places, these could be wishful prophecies, that as time goes, the future variants of SARS-CoV-2 are going to be milder, and as eventually it would be something like a fluization of COVID. Is this at all true?
Well, this comes from the belief that when the first two coronaviruses, which are now seasonal coronaviruses, crossed over into humans, they may have caused disease that was more like pneumonia. And over time, they settled down and became just the common cold viruses that we have. However, what matters for the virus itself is its ability to replicate, and therefore gaining mutations that allow it to transmit more easily is a priority if the virus was a sentient being, which it isn't. But severity is a byproduct. The virus doesn't care whether it causes less severe disease or more severe disease as long as it transmits better.
I see.
So I'm sorry, not. I don't have an answer for you. I can tell you where that came from. As there is no evidence that the virus is being driven towards less severe disease.
I see. So the fact that Omicron is more transmissible and less severe could be a mere coincidence. I mean, it could be more transmissible and more severe at the same time.
That's correct.
Thank you.
Thank you. Thank you, Gagandeep. Thank you, Professor Kang. Sushil Majumdar.
Yeah, thanks, Yuri. Thank you, Professor Kang, for a wonderful presentation. I wanted to refer to the vaccine reluctance that we see in some countries, some audiences. The thought there is that all this is happening way too fast. The vaccines have been produced very fast. They have been unleashed onto us very fast. And we don't really know the actual effect, the consequence the vaccine itself will have on our bodies, maybe slowly over time. So first question is, what are your thoughts on the rationality or irrationality thereof? And second question is, has there been any such event in the history of vaccines where people felt, oh, this is having a far too negative consequence?
So we've never had a situation before H1N1 where we tried to handle a pandemic with a vaccine. And with H1N1, the doses were limited. There also wound up being a safety event where in Scandinavia, people who had received the H1N1 vaccine made by one manufacturer with one adjuvant developed narcolepsy. So there was a concern that vaccine was withdrawn, isn't used anymore, but there were issues about, this vaccine, this product went to market too fast. After that, we've only had SARS-CoV-2. What bothers me most about the use of these vaccines, particularly in India, is the lack of attention that we pay to monitoring and to safety. These are new products. Vaccines are given to healthy people, and therefore the bar for safety, the bar for ensuring that we understand how well these products work is completely on the medical community, the public health community, the government that is giving the vaccines. And we pay a lot of attention to everything else. We don't pay enough attention to making sure that we understand how well and how safely these vaccines are working. And that's critical for vaccine confidence.
Thank you.
Thank you so much.
Thank you. Thank you, Professor Kang. And I'll relieve you at this point. There were multiple questions. It was beautiful talk. An absolute delight to hear you again. And I wish that you quickly recover from your own infection, and we have a healthy you visiting us with even more enthusiasm about all these ideas of public health. Thank you. I would also, thank you. I would also like to thank the audience for participating so generously in this particular event. And I would thank Professor Ullas Kultur for actually helping me invite Professor Kang. And also I would like to sort of request my friends, colleagues in the TIFR community that as the Omicron spreads rapidly, please also spread your health status rapidly if you are positive, because there are some not so fortunate ones in our community who can't be home quarantined, and they have to leave the campus for a BMC facility, which is not always ideal. So I would request all of us to be as much cooperative to each other so that we know that we're all healthy together, okay? So I would also like to thank people in the AG66 lecture theater for transmitting this colloquium in the lecture theater. People are interested in listening in the big screen. So thank you for doing that, Ms. Triji and co-workers. And for the next session, we have Professor Kanak Saha from Ayuga, Pune, who recently won the Bhatnagar Award in physical sciences, talking about some of the very interesting observations of deep UV photons from expanding galaxies. So I hope you can join us for this next session at 4 o'clock. Thank you all, and I will stop the recording for this